Okay, so no pre-lab. Your lab report is due on Wednesday night. At 11:59 p.m. Okay, questions? All right, then let's go ahead. I think I talked to you sort of about this before. I don't know that I did it like this, though. So we're talking about why stereochemistry was important. And when you think about like a protein is like, compared to any small molecule we'd have binding to it, a protein is this big surface. All right. And so when you think about interacting with a surface, stereochemistry is really important. So let's say this surface has a red place to bind, and it has I've got a white thing over here. So it's got a white place to bind, which I'll do like that. And then it has a blue place to bind, like that. And so if I use this was my molecule, right? This has stereochemistry because it has 4 different attachments to a center carbon. Will this thing be able to bind? You see that it won't? There's no way I can turn this to get it to bind there. So this enantiomer won't bind. Well, this one will. All right, so that's why this is important. Only certain things are going to bind to proteins, and only one enantiomer usually and not the other. When you think about drugs, when you're designing drugs, often that becomes important. I think maybe it gets more used, or whenever I hear about it from Dr. Sariski, who used to teach pharm chem, she talks more about it, how they'll release the racemic drug, so both mixture, and treat people with that, as long as you don't have bad side effects from one of the enantiomers. And then once their patent runs out, they'll then release the purified enantiomer drug and try and extend the patent on that and send their sales reps out and things like that. Even though it may not make a difference, you just take twice as much of the racemic and it's okay. But it's one of the tricks they like to play. Anyway, questions? Okay. So then let's go ahead on. We are going to be talking about, actually we ended up last time talking about doing a problem where we have multiple pKs we need to worry about, right? I didn't put the chart in here. So last time we did what? Tyrosine? Is that right? Cysteine. Did we do cysteine? That's what I have in my notes. Okay, great. So we'll do tyrosine this time. So if you look at tyrosine, tyrosine has this structure. I'm going to do it in the fully protonated state, so I'm doing my pKs correctly. It's going to have, perhaps not with bent bonds like that, but it's going to have that. And then the pKa's here, whoops, I didn't do it in its fully protonated. I said I was going to do it in its fully protonation state, and then I didn't. There we go. All right, so this pK, I'm going to make these up. This is 4.0. Actually, no, it's going to be less than that. It's going to be like 2.5. And this pK is going to be about 10 something, maybe a little less. Let's make it 9. We'll make it 9.5. All right, and this pKa is going to be about 8. And those are pretty close, although not exact, because I haven't looked them up. So you've got this problem. Then the question then could be, calculate the percentages of all forms of tyrosine that are present at more than 1% abundance at pH 4. Okay, and so for this sort of problem, what we've got is we have three functional groups that could be protonated or deprotonated here, but really only one is relevant for the percentages because the other two are so far away from what pH we're at that they're all one form. Yes?
Can we move that by any chance?
This thing? Yeah.
Yeah, thank you.
Mm-hmm.
Thank you.
Mm-hmm. You're welcome. All right, so the first thing I would do when I would see this is I would look at it and decide which functional groups are can I rule out. All right, and in all the problems I give you, you'll be able to rule out all but one functional group. And then the other functional group you'll have to do Henderson-Hasselbalch on, or you'll be able to rule out all functional groups and you'll just draw one form. Okay, you want to be able to recognize that because if I give you one of those problems, you don't want to spend 10 minutes doing Henderson-Hasselbalch when you didn't need to. So this one, pK is 9.5. Is this in two forms or one form based on our biochemistry assumption? One form, right? Because
It's greater than 2
it,
units away.
it's greater than 2 units away. Okay, so this one's one form. This one over here, two forms, right? Because we're only 1.5 away. This one here, one form. Okay. In both cases, our pH is much lower than this pK and this pK. So if our pH is lower, are they going to be protonated or deprotonated? Protonated. Your pH is lower, so relative to these pKa's, you have lots of protons around, so they stick to things. Stick. Okay? Like that. So our two forms, I don't want to lose my room here, but I'm going to. So our two forms here are going to be the protonated amine is going to be present in all forms. And then this is our form that's our carboxylic acid had the pK of 2.5, right? So it'll be protonated in one form. It'll be not protonated in the other form. Okay, don't forget your charges on these amines. Charges are super important in this class. And then our phenol group here is going to be protonated as well. So those are our two forms. All right, I might call this one HA, and I'll call this one A minus, so when I get down to the bottom, it's clear which one was which, okay, of my Henderson-Hasselbalch equation. All right, so remember that our pKa, it's almost off screen now, but our pKa, it was equal to 2.5, right? So we're going to come down here, and now we can do our Henderson-Hasselbalch. And so pH is equal to pKa plus the log of A minus over, HA. All right, and so our pH was 4.0. If you scrolled up, our pKa was 2.5 plus the log of A minus over HA. So it's going to be 1.5. I'll subtract 2.5 from both sides, is equal to the log of A minus over HA. I'll take both sides to the 10, or 10 to both sides, I should say. Can somebody help me out with that? I didn't bring my...
31.62.
What was it, 31?
0.62.
Okay, good. And I'm doing I did the mental check in my head there that it kind of made sense, right? It's more than 10. It's less than 100. Okay. And that's going to be equal to A minus over HA, like that. And so A minus, if I multiply HA times both sides, is going to be equal to, there's going to be 31.62 HA in an A minus. All right. So now I know that 100% is equal to HA plus A minus, right? And so now I can substitute in this HA, I can sub this in right this, well actually let's draw it like this. This can go there. And 100 is equal to HA, which is 1 HA plus 31.62 HA. All right, and that ends up being 32.62 HA. All right, and then if I divide both sides by that, I'm going to get about 3, right? So if I divide both sides by 32, I'm going to end up with 3. Somebody else doesn't do it. I'm just going to call it 3.3. That might not be what it is equal to HA. Do you agree with that?
3.1.
3.1? Great, thank you. Did I just miss? Oh, wait. Well, we're using the pK of 2.5 because it's the only one within 2 units. And the other ones that are going to, we're assuming if they're more than 2 units away, it is 99% or more only one form. And so we chose that form. We just said, okay, we're ignoring the little fraction, tiny less than a percent that's left after that.
Wouldn't it be 100% divided by 32.62, which would give you 0.031?
100 divided by 36.62 is
100%, right?
Yeah.
So,
Okay.
which is equivalent to 1.00, right?
No, don't make it a fraction. We're solving for percents here. You could make it a fraction and then convert it to a percent when you're afterwards, but this will give us the percent straight out.
Okay.
So if you do this as a fraction, which you certainly can, and the question's asking for percent, make sure to convert it to percent once you're done. All right, and so if 0.31% is HA, the rest has to be A minus, right? And actually, I could get rid of the concentration things right now. I leave them in up there just so it's easy for us to see where to substitute. But really, we're doing percents. They're not really concentrations. I won't make a big deal about that if you do that someplace. And so that the rest is going to be 96.9% A minus. Okay, and up above where I had the 2 forms, I did label them as HA and A minus, so I don't need to worry about indicating it because it's indicated already. Questions? Let's do the mental check real quick. Does that make sense? The pK is so we have lots more of the base form, right? The pH is higher or lower than the pKa? Higher. So there are fewer protons around, so they come off of things. So it makes sense? I can tell you that this looked funny for me. You probably don't care about this, but that looked funny for me because clearly when I make up problems off the top of my head, I'm always making them up where it's more acidic, or almost always making it more acidic than the pK question, because that looked, my head going, is that right? Is that right? That looks weird. Is it right? Okay, questions on that? All right, so we'll do that sort of thing. So you've got proteins, and proteins can have these multiple pKs all over the place. We're not going to do something like that, where you have to go through and look at all the pKs for the side chains and then rule them out. That would be a fun question, but I probably won't do it. But what you do have when you talk about proteins and them having multiple groups, you can't see the groups here. You probably can't even see the sequence, right? But at least some of you can't. This is insulin. Can't make it too big because I need to draw on it. All right, so this is insulin right here. And so a concept that we need to think about as well is something called PI. Okay, and PI is the pH where a protein has no charge overall. Okay, that doesn't mean it doesn't have charges. They just all cancel out. That makes sense? And it's even more complicated than that, although we won't have to do it. I'm going to describe it real quick. It's not that I have a plus and a minus and a minus and a plus and a plus and a minus. Remember, all of these things based on the pH are neutral or minus, neutral or plus, right? So if you take the fraction that each of those is plus or minus and you sum it all up, then it adds to 0 at that pH. Does that make sense? You don't need to know that part of it, but you do need to know it has no charge overall. Okay? And so people can think about this when they're to modify proteins to make them useful. And so this is the sequence for insulin, and there is an insulin variant called glargine insulin that is a long-acting insulin. So you take this insulin, and it's going to slowly be released into your blood over a number of hours. And it's a genetically modified insulin, and the way it was made is there have been 3 amino acid changes to it to change the PI. And it's going to raise the PI from, I think, 5.4 to 6.7. I'm just having fun because I could have looked it up. I got it right. Okay, from 5.4 to 6.7. Okay, what happens, the result of this is that when you inject, or this insulin is stored, it's in solution at pH 4. It's soluble at pH 4 and not soluble at pH 7 as a result of this change. And we're going to call this storage conditions. And not soluble at pH, I'll go ahead and I'll say 7.2, and we're going to call that muscles. It's really the blood around your muscles, right? Okay, so what happens is you inject this insulin into your muscles. It's soluble. You inject it in your muscles, it precipitates out in your muscles, and then it slowly dissolves back into the blood over the day. Okay, questions on that? See how this concept of PI and being able to change it is useful? All right, and then if you were interested, the actual changes here. All right, are that this glycine right here, this is going to get changed to a glutamine or asparagine. Ah, shoot. No, it's asparagine. So that gets changed to an asparagine. And on the end here, they extend this by 2 arginines. And that's how they get the effect. Questions on that and how that concept of PI is useful? All right, PI is also useful in that you can separate proteins based on PI. Yes?
I'm just like, so does it change because we're changing like relative charge of the amino acid?
So does the PI change?
Yeah, like I'm confused about this.
Yeah, so what you've done here, yes. Does it change because of you've changed the amino acids? So this glycine to asparagine, I'm not sure what that does, whether that is actually causing a change in the pKa. It's possible. This asparagine is right next to a glutamate, so it might stabilize the charged or uncharged form of glutamate. I need to think about that. Possibly draw it, see if we get any. It's probably too big to be like one of those special 5-member ring things, 5 or 6-member ring things. But over here, arginines. What do we know about arginine? They're charged, right? They have positive charges on them. Okay, you guys need to do your flashcards for the quiz, right? But arginines are 2 positively charged amino acids at regular pH, or at blood pH. Okay, and so that's how we're getting that change in pK or PI. And I'm not expecting. It'd be nice for you to know this story, right? It's a good example, but I'm not expecting to know the details really of it. All right, so you can use that, and what this is called is isoelectric focusing. Is the technique we use to do that. Um, and so what you do is you set up a buffer condition where you have a pH gradient. And the buffer condition is such that you have these buffer molecules that can be positively charged or negatively charged, and their pKs are such that they migrate, the buffer molecules migrate to the place where their pH would be, if that makes sense. They're going to be neutral where it's set up pH. And your proteins go in as well, so you essentially dump it all in at once, and you turn on the electrodes, and what happens is your pH gradient gets established. Let's not worry about that too much. Let's just pretend we have the pH gradient. We have a pH gradient, and the proteins will migrate until they are neutral, and then they stop moving. So unlike the gels we ran in lab where we could run stuff off, you can't run stuff off because you could just run this as long as you want until it dried out, and then it would stop moving. But you could run it as long as you want, and the proteins would move to where they were neutral, and they stop. Okay, which is really cool. But you can see it here. Let's say I have a protein right here. So let's say it's got some carboxylic acid that's not protonated, right? I move towards the positive electrode, that's a positive right there, okay? And also low pH is over here. So as it moves this way, it's getting to more and more protons around. Eventually the protons stick to that and make it neutral, and it stops moving. That makes sense? So that would be this one, I guess. And over here, it's positive. So that was a carboxylic acid. This is a protein. So this is some amine on the protein. All right. And that amine is protonated way down here. Okay. And it's going to move up until we lose a little bit more of, my functional group probably don't work for this, but it moves up until it deprotonates. We get a high enough pH that deprotonates, and then it stops. Does that make sense? It's more complicated than just carboxylic acid and one carboxylic acid and one amine. Because all of these things are some percentage protonated, some percentage not, and so you just get the mixture of all of them until we hit the PI. The same thing for all of those. Does that make sense how that works? I'm not really going to write more because it's actually this caption down here in terms of written stuff does a good job. Actually, when I was working in industry, my group did a little, we did a little bit of it. We were working with a separate company that was doing this. All right, I'm going to move on, and we're going to start talking about protein structure. All right, then there are 4 levels of protein structure. They're shown right here, but I'm going to go ahead and talk about them as well. Maybe I'll put this up by the top. You've probably heard about these in one of your biology courses, if you've had biology courses. All right, first level is our primary structure. And primary structure is just the order of the amino acids in the protein chain. I'm going to just say linear. And I'm going to put protein, and I'm going to put in parentheses peptide chain. And then I'm going to put over here, I'm going to talk about peptide. I'm going to define peptide. I think everybody knows what a protein is. A short protein. Okay, and then I'm going to maybe jump back and forth in terms of my terminology. All right, that's the primary structure. So it's just like what's shown here, which I hope you can see in back. And if you can't see in back, I can't really make it bigger and then also do this. But it's just this protein goes lysine, lysine, glycine, glycine, leucine, valine, alanine. That's the order. All right. Now that chain is going to be able to fold up into a local three-dimensional structure. All right. And that's what we call secondary structure. And it's a local 3D, so three-dimensional structure for the protein. Okay. And we'll spend a bunch of time talking about that. One of the things I think is really, really kind of amazing about this is when we think about secondary structures, you think, gosh, figure there'd be a ton of different secondary structures, right? You have so many different structures, so many different three-dimensional structures of proteins. You'd have a lot of secondary structures, but there's really just a few major ones, all right? They might have slight variations, but we have alpha helices, we have beta sheets, and we have turns, and that's about it, okay? We're not simplifying much for this class in terms of that. All right, so the next thing is going to be our tertiary structure. And so here I'm going to blow it up and then I'll shrink it again. This is like our tertiary structure. What they're doing is we're working our way up to hemoglobin over here. But our tertiary structure is a three-dimensional structure. It's the secondary structures coming together to make the three-dimensional structure of a complete, here it's talking about a polypeptide chain. We could talk about a complete protein. Okay, we might talk about it as being a protein subunit. So tertiary structure. All right, that's the global structure of a protein. Let's say a single protein chain. All right, so the secondary structures come together to make that tertiary structure. And so if we look up here, if we're talking about doing hemoglobin, we're going to have a bunch of alpha helices. There's our alpha helix, and there'll be an alpha helix there, and alpha helix there, and yes, 2 there, and then 1 there. Okay, and those alpha helices are going to be, there's going to be an alpha helix, and then we might have a turn between them, or we might just have a loose undefined sequence, and they're going to all fold together to make this structure. Okay, staying in those secondary structures. And then finally, we have quaternary structure. Quaternary structure. And what this is this is multiple protein chains coming together to make kind of the full functional protein. So for hemoglobin is a wonderful example of this. And so in the case of hemoglobin, we have 4 protein subunits, and they come together, and they're not bound together by like any covalent linkage. They come together, they all stick together, and then in terms of quaternary structure, the magic is that they then talk to each other usually. So we'll talk about how they talk to each other, but it's probably using charges. But that's okay. But it's probably they're gonna use differences in charges to talk to each other, so one knows what the other one's doing and reacts to it. It's really it's magic. It's cool, really cool the way it all comes together. It's not magic. We can explain all of it. So that's what quaternary structure is, though. Questions? Okay. So how do we know what these structures are, right? So I can do this really cool thing. You could try this as well. You don't even need a special tool for it. So if I go and I go protein data bank, protein data bank actually has a visualization tool on it. Ooh, molecule of the month. So what is that? Can I even spell that? Abscisic whatever receptor. We'll copy that up here. We'll see what we get for that. Okay, so here is one of those. Crystal structure of the highly affinity abscisic receptor bound to something. Great. Let's go look at that. So we can come over here and we can look at the 3D structure. And they have a viewer, which is just really cool. I use this when I'm like in meetings and we bring up something comes up and I need to look at the structure real quick. The stuff I do at VT, I'm the chemist in the group, and everybody else is a physicist or a computational person. So they ask me chemistry questions. And so what we can see here, this is this structure. It's this beautiful structure with alpha helices and beta sheets all folded together. The red dots are waters. We can hide them. Let's hide our waters. But it's really just very cool what you can do with these sorts of things. Anyway, any questions on that? I forgot even why I started that tangent, so we'll go back to the lecture. Yeah, and it looks like, hey, you know, it's still recording. It's still recording. I thought it always stopped recording when I changed screens. Okay. Anyway, so how do we know what these structures are?
We all had trauma from HMR.
From what?
HMR.
Oh yeah, this is cool. All right. Anyway, how do we know what these structures are? All right, so how do we know these structures? Most of them are not going to actually be from NMR, but some of them will be. And so most of these, I'm going to say mostly X-ray crystallography. All right, so what is this? This is a double slit pattern. I didn't have time. My office, as you've seen, is a mess of my stuff and Dr. Saristi's stuff, and I keep needing to throw a box out a week. I haven't managed to keep up with that yet. But one of these days I'll find my laser pointer, and I could show you this, right? So what I could do is I could take a piece of aluminum foil, and I could take a razor blade, and I could make two little slits on it with a razor blade, with my glasses on. And then if I shine the laser through it, the laser actually makes this sort of pattern. Anybody remember what that is from physics?
It's because there's like waves,
Uh-huh.
and they overlap and they interfere with each other and create a pattern.
Right, exactly. So it's just like this, okay? So if I shine these through, what's happening is light acts as a wave. It's a wave, it's a particle. It's whichever one you want that best explains the situation you're in. And so we can imagine we've got my waves of light coming in here. And what's going on when we think about that for waves of light is this red light is going to be waves, right? Just like waves in the ocean. And it's a laser, so all the waves line up better than what I've drawn here. All right, and so this is maybe the top of each wave here. And so those waves hit this piece of aluminum foil, and then they spread out. Just like if you saw waves crashing against, when they crash against something with a hole in it, you're going to get these, they spread out in circles like this. And when they spread out in circles like that, you're going to have places. So this was places where the waves, the tops of the waves came together. And so they made a bright spot. Here, tops of the waves come together, make a bright spot. Same sort of thing here, a bright spot. And then in between, you have places where you've got right here, I've got places where one wave is there. See how this wave is kind of in the middle of these two tops? So that's a top on a bottom, and the top and the bottom cancel each other. Does that make sense? So in other words, I have right there, what's going on is I've got one wave doing this. I'm going to erase this one. And then the other wave, right there, they're exactly lined up so they cancel each other. And so if I add those two together, what I'm going to get is that. Does that make sense? They cancel each other out at those spots. So that's where our dark spots are, and our light spots are where two crests meet each other. And if two troughs meet each other, that's what these ones are not showing are, I think. Okay? See how that is? All right, so what you can do with this, though, I don't know if you have a physics lab. Presumably you have a physics lab where you do this. Yeah? No? You know,
I think it's like you have to take like 2 or 3 physics classes to do this.
okay. So, but if I know how far away my wall is that I'm projecting these on, and I know what the spacing of the slits are, and I know what the wavelength of, watch, let's change that. If I know what my wavelength is, okay, and I know how far away my wall is and what my pattern is, I can then figure out what the spacing of those slits is, okay? Not very exciting. I could have measured it with a ruler, right? Because I made the slits. But imagine now the slits are you've got a crystal of a protein, and the slits are the spaces between the atoms. Okay, I can't measure that with my ruler, but I can shoot X-rays at it, and it will diffract the light just like this happens, and then I can back calculate where the holes were. And if I know where the holes were, I know where the atoms weren't, right? And I can figure out where the atoms are. Does that make sense? Okay. So that's how this works. And so this will talk about actually what happens here. So right here, what we've got is somebody's crystallized a protein. Okay, so they've got a protein, it's all packed uniformly. So the holes make a uniform pattern, and they put that protein there on this little rod hanging out there, and they shoot X-rays at it, and they get a pattern of spots behind it. Okay, so A is shoot X-rays at a protein crystal and observe the diffraction pattern. Okay. You then use the pattern to create an electron density map. Okay, and so essentially what's going on here is there are no electrons here, right? So my X-rays, I back calculate from this pattern where the holes are. Well, there's some holes, and there's some holes, and we're not showing there's more protein around this, right? But I can figure out where there are holes, and where there aren't holes, there are electrons. And where there are electrons, there are atoms. Okay? So that, yes.
How do you get that from that picture?
How do I get this from this? So I'm going to use a fancy computer program as the easy way to talk about it. I took a class in grad school where we learned how to do it by hand for smaller molecules, but I've forgotten all of it. But really, it used to be. Now it's something that computers handle most of it, and hopefully the people that are super expert in. There's a lot of people that would just do this and use the tools and don't exactly know what's going on, but there's a lot of calculations involved. But I seriously don't. I got an A in the class, but I don't remember any of it. So you get this. So then you're going to use another fancy computer program to take. You've got a structure, right? You know what atoms are kind of out there. You know what possibilities are, and that's going to take your structure and it's going to try to fit it to this map. Okay, so I'm going to say fit the atoms you have to the map. Okay, remember, you know what the protein sequence is. You need to know what the protein sequence is. Here it is. Okay, this isn't a great example for showing you that because this picture, they're actually showing you the heme, right? That doesn't have the protein sequence, but they're going to have electron density map for the whole thing. And you know if your protein sequence, if you've got a really good fit, hey, this glutamate fits really well here, and I have a lysine right next to it, and the lysine doesn't fit at all, well, I've got things in the wrong place, and I need to try something else. Does that make sense? So the computer's going to try things, and it's not going to try everything. There's some algorithm that makes it smart the way it tries things, because otherwise it would take longer than, and I'm not kidding, it would take longer than the universe has been around to solve. Okay, so you have to, the algorithms have to be smart as well. Okay, so fit the atoms, and then, well, D is the same as C. D, more of C above. We'll just say the final structure. Questions on how that works in general? So once again, we've got the diffraction of light to give us a pattern. That'll tell us where the holes are. From that, we can get an electron density map. With electron density map, we can fit our molecules that we know we have in there, and then from that, we build that up to the whole structure. And generally, once somebody has done one protein structure of a family of them, the rest become really easy because they generally have similar structures. Okay, NMR. How do you solve structures using NMR?
By looking at the peaks and the integrations and a whole bunch of other stuff.
Yes. I'm going to say something more specific than that, but how do you solve structures using NMR? Okay, so essentially the big technique here, and you use other techniques as well, but the big technique is nosy. Did you guys ever do any nosy? I think you don't. Yeah, it's not cozy. Okay, so we're going to say you use nosy. Okay, nosy, which is the nuclear Overhauser effect, which gives you information about what protons are near other protons in three-dimensional space. And the trick here is this is not through bonds. Not through bonds. All right, so in other words, if I have, let's say I need something non-exchangeable. Let's say I've got my protein sequence here, and it kills me to draw actual hydrogens. But I've got a valine here, and I'm going to have to fix my sequence, give myself a little more room. And then over here, let's say, let's make it easy on me, I've got an alanine, okay? And because this protein sequence curves around, even though these two things aren't close to each other in the primary sequence, they're not next to each other. They're maybe, if that was spread out, they're 10 nanometers away from each other. Yeah, sure, 10 nanometers. 10 nanometers away from each other. So you wouldn't be able to see interactions. Because it curves around, I can get that I have an interaction here between these protons and these protons. And so I can say, well, those things are going to be, 5 angstroms away from each other. I don't know what the exact limit is, Brennan. Okay? Does that make sense? All right, so, and the way I figured that out, so this is like the structure of a protein. This is like a 1D, okay? Dr. Sariski did this in college, not college, grad school. But what you do is you actually, this is where you would use like things like COSY, is that you figure out what one of those protons are, and then you kind of walk your way down. And you're going to use 2D, you use COSY to do that. But then you have stuff like this that you can use to figure out where things are close to each other. So this is, what we're looking at is protons versus protons here for chemical shifts. And then if we get a peak here, that means that this one is close to this one. Does that make sense? And we do that over the protein and we get a bunch of those. I mean, we don't need to get a tremendous number of them, but we get a bunch of those, let's say. All right. And then what you do is you put them into a computer model. You're going to do molecular dynamics minimization here. And so you've got your protein, right? And you say, okay, I want you to take this protein and I want you to try and make it lowest energy possible. But you also have to make sure that this proton here is close to this proton here. And this proton here is close to this proton here. And this proton here is close to this proton here. And by doing that, you restrain the system such that the minimization programs are good enough that they generally get it right. Okay, what they're showing you is the minimization. Here are a bunch of different possibilities, right? That's why this isn't one. X-ray crystallography is the same thing. This is just showing you uncertainty, right? X-ray crystallography stuff, we see one position for everything, right? But there is uncertainty there. Yes? That's a great question. So what can you do with knowing the shape of the protein? If you know where certain residues are, you can then like make drugs to help you work on those proteins. So here, let me get us another one. All right, so I'm going to do 3UO4, which is one I work on it's one that I work on in my research. There's a whole bunch. I just recognize this one, or just remember this one at the moment. What is this? A transferase inhibitor. Let's do one I think I remember, 4GID. Let's do that. This one's related to Alzheimer's. All right, so this would be an inhibitor that related to, you've heard of it, you get these precipitating proteins, right? So what happens is they get chopped a little bit, it makes a hydrophobic piece, and they all stick together. Okay, so this is an inhibitor that would keep them from being chopped. I know this one because there was a lot of drama around it in the group. Not really, but we had to adjust it because it doesn't deal well with things. So here, this thing in pink, can you see this thing in pink right there? That's an inhibitor they're thinking about using with it. Okay, so actually what I could do is I could let you see. So there's the inhibitor. This is just some buffer they use to help get the protein to crystallize, so we can ignore that. But there's our inhibitor there. You can see it has some fun things on it, like this is like a sulfuric, it's like a sulfuric acid, or it's a sulfur amide. It's at least kind of the way it acts. That's not important though. To answer your question, I mean, it is important for outbinding, but if I know that this is the structure of my protein, I wish I could easily color them differently here. But if this is the structure of my protein here, I know that this is my binding site my inhibitor might fit into. And right now we're cartooning the protein, right? Because if I showed you all the atoms, it's just a mess. You can't see stuff. But if we don't cartoonify the protein, then I could also see what functional groups are everywhere like that. So I could see what do I think, I could look at it and I could see what do I think will fit well in there. And organic chemists use a design drugs to say, oh yeah, I think, I could build this, I could build that would fit that well. Okay, and from the perspective of the work we do, or what I do at Virginia Tech, we look at these things and we're modeling how tightly does this bind, is what we try to model. So that's where the structures are useful, because we use these structures, when we do molecular modeling, we don't start with a spread out protein. We start with this structure, and then we see what happens. So that's how knowing the 3D structures is useful. It can also explain how things work. Yeah.
It's 5 minutes.
Oh, yeah. Thank you. Okay, so you saw how the NMR stuff worked there. I'm not really going to make you